This is where we left off, right?
Yes.
Did somebody just say Happy Friday?
We finished this.
Oh my God. All right, so this is the last slide we did. We talked about intrinsic factor. It's released from the cells in the stomach, parietal cells. It carries B12. B12 is released in the, what is happening? Where did I go backwards? It is Friday. It's released in the ileum, and you get vitamin B12 absorption. So keep in mind these things right here, this and these, the ileal disease, the resection, the stomach problems, and what can be caused by lack of B12 is going to be important. All right. How do we regulate gastric secretion? All right. We have nervous system and the endocrine system. All right. Everything on this slide is on your sheet that I just handed you. Somatostatin inhibits acid secretion. Way you can remember that, statin stops. Okay, a statin stops. So that inhibits acid secretion when you have too much. Acetylcholine would be a so I lied. That's a neurotransmitter. Through the vagus nerve, stimulates gastric juice secretions, specifically acid, and the secretion of a hormone. So you have nervous system secreting both gastric juice and a hormone that will aid in secreting gastric juice. Okay? And histamine. Histamine, we know it as that thing that causes allergies, but it is also released by cells in the stomach, and it increases gastric secretion, particularly acid.
Real quick, did you say somatostatin is a hormone?
Yes.
Yes.
All right. Specifically, acetylcholine, again, that's nervous system, acid, mucus, digestive enzyme, this would be the inactive form, zymogen, and gastrin, which we know is a hormone. Histamine, like we said, the acid. Gastrin, again, the acid. And gastrin is going to be the most important stimulus for acid secretion. I just want to make sure I'm 100% sure I'm recording. Yes. Okay. So, role of vagus and gastric secretion. What does that mean? Role of the nervous system in gastric secretion. All right, we know that stimulating that increases acid secretion, but how does it do it? One, it directly stimulates the enteric nervous system, the ENS, if you will. That will then stimulate gastric glands, which then directly stimulate acid secretion. Okay, so that is a direct pathway. But we also said that it will go to other tissues, such as endocrine tissue, this thing called the pyloric gland, which will then secrete gastrin, and gastrin will go to secrete acid, or I'm sorry, increase acid secretion. All right. Atropine is a drug that blocks parasympathetic pathways. All right. If we apply atropine, we get rid of the direct pathway. The indirect pathway is unaffected. Why? Gastrin-releasing protein is the neurotransmitter. If you block acetylcholine, it's not going to block GRP. Therefore, atropine you'll still have acid secretion through the indirect pathway, even though you've blocked the direct pathway. Does that make sense? And the reason for that, again, different neurotransmitter that does not get blocked by that same drug. That's like evidence that these are two separate pathways. All right. Here is kind of a schematic of how that works. So everything on here I will put to you was covered right here. So we have the vagus nerve. Here would be your indirect pathway. I'm sorry, this is just the indirect pathway. Here would be inhibition of that indirect pathway. Okay, so vagus nerve, acetylcholine to this endocrine cell. Okay, that will release GRP, go to the gastrin cell, release gastrin, right? And of course, that will go to release more hydrochloric acid. All right, that can go through the vagus nerve. What can also stimulate this gastrin cell to release gastrin to increase hydrochloric acid secretion is peptides in our gut and amino acids in our gut. Okay, so that what does that? That tells us that our body has a bunch of proteins coming in. We want to secrete more acid to help digest these proteins. All right, vagus nerve can also stimulate the somatostatin cell. Somatostatin goes to directly inhibit the gastrin cell so you don't secrete gastrin. So somatostatin stops acid secretion by inhibiting gastrin from being secreted. All right. The other thing that will stimulate the somatostatin cell to prevent acid secretion is acid. All right. So the thing here is when you have too much acid, and how would you know if you were in a laboratory if you have too much acid in a beaker? How do you measure acid? pH. So what would the pH be? Too low. Okay. When it gets below 2 to 3, you start inhibiting acid secretion because you don't want it to get more acidic. Why would being more acidic be bad? More destructive, right? Okay. So just to explain this one, here's your G cell that will release gastrin. The arrows are going this way. It'll go into the blood, so that's really important to know that it goes into the blood. This is not a paracrine substance. It'll go to our ECL cell that we've described and cause the release of histamine that will stimulate parietal cells to release acid. All right, so that's the pathway gastrin goes to release acid. Gastrin can also directly go and bind to that parietal cell and cause acid release. All right, so 2 pathways you can go through to get release of acid from a parietal cell, through stimulating histamine release and through directly stimulating acid release from parietal cells. Does that make sense? Okay. All right. When our stomach is empty and we're secreting acid to maintain things, pH is about 2. All right? And that's 10% of the maximum amount that can be released in response to a meal. All right? So when our stomach's empty, we are producing acid, but that's only about a 10% rate. Once we get food in there, we want to secrete more acid to make sure that that pH stays low. All right? What do you think happens when you put food in your stomach to the pH? pH goes up. So you want to secrete a lot more acid to make sure that that pH goes back down so that the appropriate enzymes can be activated. All right, so what stimulates gastric secretion? This is coming more from the neural pathway, which we just outlined how that interacts with the endocrine pathway. We have the cephalic phase, the gastric phase, and the intestinal phase. All right, what does that mean? The stimulus for acid secretion occurs in the head, occurs in the stomach, and occurs in your intestines, specific places. All right, this is showing all 3 phases. So the cephalic phase operates through the vagus nerve, and that is what we are seeing right here. Okay, remember, gastrin and acid is both secreted by the stomach, so that means the vagus nerve is going to go to the stomach to stimulate the release of these substances. The gastric phase local nervous secretory reflexes. So there are certain things in there happen in the stomach that stay in the stomach, stimulating acid release. Vagal reflexes, of course, that would be increase in pH. That would stimulate the sensory vagus nerve. Then the motor vagus nerve would come down and stimulate acid secretion. And gastrin histamine stimulation, which is, this thing we just talked about right here. All right, then the intestinal phase. We have nervous and hormonal mechanisms. We'll get into that when we get into that to that section. So, cephalic phase. Think about this. Here I'm going to give my pot roast lecture. For some reason, I absolutely love pot roast. You like put it in there. You put the roast in there, and the carrots and the potatoes and the onions. Oh, and then you put some onion soup mix in there, and then some beef base. Oh, and then you come home, and then you set it in there, and you're like, this is going to be good. And then you come home from work, and you're like, it smells so good. So, and I'm thinking about it during the day. When I'm thinking about it during the day, you guys think about something that you really love to eat, and it makes your mouth water. When it makes your mouth water, notice your parotid glands will actually start tingling. It's the weirdest thing. I love it, though. And then you get home, and you smell it. That same thing starts happening. All of these are related to the gastric phase of secretion. Your brain is saying, oh, we're going to eat something really good, and it's preparing your stomach for that. Okay, so it's going to start secreting acid. All right, that's about 30% of acid response to a meal. Okay, even though we say it's a cephalic phase, mechanoreceptors are involved. That's because when the food touches our lips, that's still considered part of the cephalic phase. Chemoreceptors, that smell, taste. Central pathway, when we think about food, conditioning, and hypoglycemia. We're low on sugar. It's going to tell our body, all right, you better prepare for some food because we're going to make this guy want to eat. All right, all of this can be blocked by a vagotomy. All right, so if they cut my vagus nerve and I think about that pot roast, my stomach won't get activated. All right, what's it going to do for my digestion if 30% of secretion comes from cephalic phase?
Slow it down.
Yeah, it'll slow it down. It'll severely impair your digestive ability. Particularly, what macromolecule starts digestion in the stomach? Proteins. So it's going to curtail protein digestion, which, if we all know about survival, protein is the most important one, right? If we don't have protein, we die. Gastric phase. This is where most of our acid secretion comes from, all right? And that's when the food enters the stomach. So we have that peristaltic passing down through the esophagus, and it goes bloop, and hits that acid bath. Okay, so enters the stomach. The stimulus for this phase, increased pH, because we've already said that when you put food in the stomach, pH is going to go up because you're buffering that acid. Distension, when you put food in your stomach, what happens to it? It gets bigger. So this would be a later phase unless you are the kind of person who just would like stuff a whole hamburger in your mouth and it goes down. My son did this the other day. When there are peptides and amino acids, so proteins, okay, because, and I'll put it to you this way too. Protein digestion starts in the stomach, and that's the only chemical digestion that occurs in the stomach is proteins. None of the others, just proteins. And also these substances will also increase acid secretion when they're in the stomach. So when you drink that glass of wine or you drink that cup of coffee, that's preparing your stomach for food, even though food's not coming. So it makes it real acidic. So when they say this stuff is really bad for your stomach and causes ulcers, that's how it increases acid secretion. That's why when you go for that cup of coffee, make sure you have that crepe with it.
I understand like the wine and the coffee. Can you go into more detail about the calcium?
Oops, going the wrong way. Because calcium is, when you stimulate these cells to secrete acid, one of the things you do is open calcium channels to allow that the secretions to happen. So when you add excess calcium, that's going to just stimulate it even more. You're providing more extracellular calcium to stimulate the cell to secrete more. All right, then the intestinal phase. That only counts for 10% of the response to a meal. And the intestinal phase, once the food gets in the intestine, like, you should be done secreting acid, right? But it's going to come in phases. So this 10% would start like when the first little bit heads into the duodenum, and then it'll say, all right, we've probably got more to digest. Let's secrete some acid to finish that shit up. Yes. All right, so stimulus for this, distension of the small intestine, so it will bulge, secrete acid, and amino acids, just like in the stomach. Okay, this graph looks intimidating. It's really not. And there will be a question about this on the exam. Here's your meal. That's where you eat your meal, this point right here. This is time. This is pH. Okay? The green line represents how big your stomach's getting. The blue line represents the pH. And the red line represents how much acid is being secreted. All right? So, what happens to both pH and the volume of the stomach when you first eat your meal? It goes straight up, and it goes up really quickly, right? So, right away, you have an increase in pH and an increase in volume. Both of those, what do we know do in the stomach? They are stimuli for acid secretion. So, when these start going up, you're going to start secreting acid, but because these have to happen before the acid secreted, that's why the curve of the acid is a little bit slower than what you see with the increase in volume and pH. All right? As that acid increases, what happens to your pH? It starts going back down. Okay? What happens to the volume? It starts going back down, right? Because you are actively digesting things. All right? So, peak volume and pH increase within about a half hour. Peak acid secretion, about an hour and a half. Okay? So, I'm going to put it at, here's a really important question. When is your pH of your stomach going to be the lowest?
When you're preparing to eat.
No.
When the pot roast is cooking.
Oh, I'm sorry. Yeah, did I say the lowest?
Yeah.
Yes, the lowest. Say that again.
When your pot roast is cooking.
When your pot roast is cooking. When your stomach is empty. Somebody said starvation? Or was it you? You? Okay, starvation. When is your pH going to be the highest? 30 minutes. Right, 30 minutes after you eat. All right, so knowing how to interpret that graph and what's happening at pH is going to be important for one particular question on your exam. All right, so I'm going to skip through this because we just talked about it with the graph. Early in the meal, acid secretion increases because you have the cephalic phase, your pH is rising, and you have more food in your stomach, that stretch reflex. Late in a meal, acid secretion decreases because buffering capacity of food is saturated, so your pH is starting to go back down. As the pH falls, the increase in acid inhibits gastrin. If you inhibit gastrin, you inhibit acid secretion. Good. All right, so that's going to help with maintaining proper pH levels. And then the chyme, which, as we said, that's the mixed up food with digestive chemicals. entering the duodenum. That's what that means, not the duo, the duodenum. I don't know what that might. That's not something I do. I probably accidentally hit erase, and that's why I ended up that way. Inhibits acid, entering the duodenum further inhibits acid secretion as well. And we're going to talk a little bit more about this as we go in one particular reflex that's associated with late meal. Okay. So this is a chart. Question.
I thought you said that you secrete more acid, 10% of acid, when food enters the duodenum.
Right. Only 10% is secreted when it gets to the duodenum. So you go from 60 down to 10.
Oh.
Yes. So, yeah, good question. All right, so all this is showing you is parts of the stomach. What is a stimulus for inhibiting acid? How does it work through? And does it directly inhibit or does it indirectly inhibit? Okay? So all of this I'm not going to go over this. It's a summary of what we talked about. Okay? So one of those quick looks like, all right, I forget what's happening with acid secretion, inhibition, go to that chart. All right, once we have all this gastric juice in the stomach, of course, we're going to mix it all together in that paste we call a chyme. And then peristalsis is going to, and of course, gravity is going to make sure it's all down at the bottom, but peristalsis is going to push it towards the pyloric region and this pyloric sphincter right here. Okay? That is closed. When a sphincter is closed, what's another word we use for that? Increased contraction, also increased, begins with T, rhymes with moan. Tone. Increased tone. All right? So, at least we didn't rhyme with shunt again. Okay, so that's going to keep shut until it gets the stimulus to open. And the stimulus to open we'll talk about in a few minutes. But we want that closed. Why do we want that closed? Because if it's closed, we can mix things really well. Okay, remember that washboard effect. That's weird. All right, so like I said, the chyme must enter duodenum at an optimal rate, so we regulate when that pyloric sphincter opens. When the chyme gets to the duodenum, is the pH high or low? It's really low. It's acidic. All right? But we need the pH of that chyme to be 7 in order for the digestive enzymes from the pancreas to work. So there are things that happen in the duodenum from the pancreas that will create that pH of 7. But if we dump too much from the stomach in the duodenum all at once, we can't get to this, and therefore digestion is hampered. So that's why we regulate how quickly food enters the duodenum. All right, plus we need to have nutrient absorption as well. All right, so what are the factors that will open that pyloric sphincter and promote gastric emptying? Increased tone of the orad stomach. Remember, the orad stomach is this region towards the mouth. All right, increased tone, so that means it's going to squeeze. Forceful peristaltic contractions, which we see here in B. All right, so it's going to squeeze the chyme toward the pyloric sphincter. Where are these contractions occurring? More towards the caudad region, right? So, because that's where you want to push. If you push from up here, you're pushing everything down, but here you want very precise contractions, peristaltic, assembling it that way. All right? At the same time, we're going to have decreased tone of the pylorus. All right? Decreased tone means it's opening and absence of mixing contractions here. All right? Because if we have mixing contractions here, what's going to happen to some of the chyme? It's going to go back. We don't want that to happen. So this has to be inactive for a period of time while this is opening and being pushed towards it. Okay? That's not the same as receptive relaxation, but it's a similar concept. All right? Factors that decrease gastric emptying, because once you empty, you want that to close back up. So you mix things back up again and achieve proper pH in the duodenum. So once the chyme hits the duodenum, you're activating receptors that trigger what's called the enterogastric reflex. Entero meaning intestines, gastric meaning stomach. That means this reflex starts in the stomach, sensory stimulation, and starts in the intestines, entero, starts in the intestines, ends in the stomach with a motor response. So we'll go over what the enterogastric reflex is, but what it results in is you have a relaxation of the stomach, so you're not pushing anymore. If you're not pushing anymore, you probably don't want these peristaltic contractions. If you don't want food to exit, you're going to increase the tone on the sphincter, closing it back up, and then you're going to want to start doing these segmental contractions in the duodenum to mix things up really well with the bicarbonate-rich fluid that's coming from the pancreas. Yay!
That sucked.
Yes, everybody clap. Was it security or was it maintenance?
I don't know.
You don't know? It's on the recording. If there happens to be a decrease in performance for this exam, I will look at that carefully for. If it's anyway. If it's something beyond your control, that's my fault or the school's fault that is inhibiting your performance on an exam, I can't punish you for that. Okay? So that's just something I've adopted long term. All right. Here's how that enterogastric reflex, but I'm glad you're out. I'm really glad you're out.
Same.
All right. 1. What's happening as the duodenum fills the chyme? Distension. All right. Sensory receptors here detect that distension. Remember, almost everything in the digestive system is related to stretch. You stretch it out, you're going to send sensory impulses up to the CNS, all right, the medulla. All right. That's going to process it, send vagus nerve impulses back down to the stomach, all right, started in the intestine, ending in the stomach, and tell the stomach to decrease activity. All right, it's going to tell the stomach to decrease activity. It's going to tell it to stop pushing. It's going to tell it to stop doing peristalsis. And it's going to increase tone on this sphincter. That's going to slow emptying. Okay? Then all you have to do is deal with this volume right here. Okay, so what are these receptors in the intestine? One of them we know are aside from the stretch, high or low osmolarity, which means food's really concentrated or liquid's really concentrated or not concentrated enough, acid, fat, and protein. Okay? If you start having all of those come to the small intestine detects that and says, whoa, whoa, all right, we got to digest this, slow down. All right? And then the enterogastric reflex is triggered. All right? if you have specifically fats and proteins, cholecystokinin is released. Cholecystokinin, that's on your chart. CCK. Cholecystokinin, one of its job is to increase gastric distensibility, which decreases gastric emptying. Okay, so if you had, what does your chart say is the function specifically?
Inhibits gastric emptying, stimulates. We have a chart.
Okay, so here it decreases gastric emptying.
Chart.
You need a chart. Need to put what on there?
Thank you.
That's on there. What I want you to know is on there.
Okay.
Okay, so CCK decreases gastric emptying. We'll get there. 'Cause we're talking about the stomach right now, so that's why we're staying with that. Acid, increase in acid, also decreases gastric emptying as well. So you have an increase in fat, CCK triggers it. Acid will trigger a neural reflex. All right, so not through CCK, through a neural reflex. So that would be part of the enterogastric reflex. And involvement of other hormones that are not well understood. So when you see things like this, it means they're probably being studied. We just don't know enough yet, and that's fine. We don't know everything yet. All right. So we've talked about the stomach. We've talked about everything that's secreted into the stomach. All right, acid being a big one. But acid, what's that important for? Why is that important? One major thing. What else gets secreted by the stomach besides mucus? Who said it? What? No. What's acid important for? Why do we need acid? Pepsinogen. That's why we need acid. We don't need acid to necessarily digest anything. We need it to activate pepsinogen. Acid helps with some stuff, but it activates pepsinogen and kills bacteria. All right, so we have this really acidic environment that has a lot of digestive enzymes in it that break down proteins. Remember, pepsinogen is a protease, or pepsin is a protease. Pepsinogen is the inactive form. Why doesn't the stomach digest itself? One of the reasons we talked about is what? Before I even go into anything else. Mucus. All right, our stomach is lined with mucus, so it doesn't digest itself because we have that barrier between the mucus and the mucosal cells, which we refer to as a gastric mucosa. Those are the mucosa cells. But the gastric mucosa also has protective factors in and of itself. So anatomically, the cells that line the gastric mucosa have tight junctions in between them. Okay? They have cell membranes and tight junctions, so nothing can get past it. Not even acid. All right? Physiologically, if these cells here come in contact with hydrogen ions, these cells will transport them back into the lumen. So if hydrogen ion actually does get into the cell will say, nope, go back. So it actually is actively pumping hydrogen ions outside of themselves if they happen to leak in. And we have mucus secretion. Okay? So those three things, tight junctions, active pumping of hydrogen ions back into the stomach lumen, and mucus secretion. All right. are these protective factors foolproof? No. What happens when they don't work? Ulcers. That's the big thing. Ulcers happen. All right? So, in most healthy people who eat a moderately healthy diet and don't drink too much alcohol and exercise and do all that, or even you guys, what I'm saying here is that when you're in PA school, like, damn it, I don't have time to exercise as much as I want to, and I wish I had time to eat healthy, but I'm just going to wrap in this cookie right now. So, I'm not making fun of you. There are certain things that weaken these defenses. One of them is H. pylori. Does anybody know what this is? It's a bacteria. All right? Now, you can get an increase in H. pylori if you're, like, wading around in human waste, which can happen. I mean, what happens when you go to these beaches that say high levels of contaminants in them? Most of it's E. coli, but some of it is H. pylori as well. Human waste. But every one of us right now has H. pylori in our stomach. Studies have shown that every human has H. pylori in our stomach. Why doesn't it bother us? Because we are keeping it at bay. Bacteriostatic. Thank you. Okay. So, aspirin can also weaken that barrier. I'm not going to go into why, but there's a really awesome video that shows how that happens at the molecular level, how that happens, so I recommend watching that. Ethanol, NSAIDs, through the same mechanism as aspirin, bile salts, and stress. All of these. When I said even you guys, you're not stressed for Monday, are you? No. See? Good. I'm glad to hear that. No, you didn't. You were right here. It just happened.
Right.
All right. So all of these things can weaken. If you weaken, first thing that's going to happen, gastritis. Anything that ends in itis means inflammation. So inflammation of your stomach. It becomes red. It becomes sore. You start having pain, nausea, could bleed if it gets into blood vessels. Loose stools can happen, okay, because you have these irritating factors. So if that goes on long enough, then we end up with ulcers. And ulcers can include all of these things. These can be occurring at the same time. But ulcers, the problem with those is they can perforate, right? They can perforate right through your stomach. And if you remember, Victoria has a perforation from her stomach into her jejunum. If you didn't find that yet, I recommend looking for it. It's kind of cool. When you have ulcers, mostly a lot of pain, but it can lead to, especially if you don't have a fistula with your small intestine, but it goes into the peritoneal cavity, then you've got really big problems. You have peritonitis, and that can be life-threatening. Okay? Things that strengthen the mucosal barrier, more mucus. What's this? Bicarbonate. We buffer it. All right? Bicarbonate secretion. Gastrin. Prostaglandins. Epidermal growth factor. And so on. All right? Gastrin and epidermal growth factor, things that we didn't talk about, but they are on your list. They promote gastric epithelial growth. That's how they protect. All right, you have a healthy barrier, no ulcers, and we are fucking happy. No. When I was in grad school, my PhD, my PhD, like 3 pots a day. It was like, because I was there from like 6, 7 o'clock in the morning to like 8, 9 at night every day. And I had to make sure that I was well hydrated.
You were like 36.
Do what?
Like, 3 cups or 3 pots?
3 pots.
So you were drinking like 36 ounces of coffee a day. Okay. Sorry.
Yeah, it was
And you didn't get like heart palpitations?
probably.
Okay, cool. Do you?
Yeah, I was not healthy. Probably. I also, in graduate school, I don't recommend this as a coping mechanism, but I drank a lot too. Okay. All right.
It's a coping mechanism.
It is. It's also, never mind. We won't. All right. So how do we get this ulcer? All right. Hydrogen gets into the mucosa. All right. That's the forerunner to gastric ulcer. We know it pumps some out, but if our gastric mucosal cells are damaged, they're not as efficient at doing this. That decreased cell pH leads to cell death, so those mucosal cells start dying. Mast cells that are lying underneath those mucosal cells start leaking histamine. Histamine not only brings more blood to the area, but it also secretes more acid. So that's a problem. And then it becomes a vicious cycle where eventually you have more cell death and more cell death and more cell death, and that's when you get that ulcer and that divot in your stomach. Okay, not good.
So do you always have to have the vicious cycle for somebody who's like on NSAIDs or something? If they stop that, will they gain back the strength in their barrier, or it would just stay damaged?
When something is allowed to heal and it's a big enough area, you can get some protection back, but most of that will be scar tissue, and that's never as strong as what the original barrier was. All right, think about scars on your skin, for example. When you get scars on your skin and you go out into the sun, if you're a light-skinned person, do they tan? No, which is an example of how they don't have the same function as your normal skin cells do, that protective barrier. So if they can't tan, that means they're probably not doing other things as well. All right, thing about peptic ulcers. This is really brief and easy. If you have gastric ulcers, which are where? In the stomach, it's usually a lack of mucus. That's the problem. It's usually a lack of mucus. That's the problem. If you have a peptic ulcer in your duodenum, which is the first part of the small intestine, usually it's a problem of too much acid. All right? So in the stomach, not enough mucus. In the duodenum, too much acid. Because one thing that's a big difference between the stomach and the duodenum, more mucus, less mucus. So this already has less mucus, so if you add more acid, it's not going to be able to protect itself. All right, now we're in the small intestine. Yay! What? 74. All right, small intestine. You guys know from anatomy, we have the C-shaped loop of the duodenum. Then it goes into the, most of the rest is the jejunum. And then this terminal part leading into the large intestine is the ileum. And what's one really important thing that happens in the ileum? That allows for what? Absorption of what? B12. B12. Good. Okay. Mostly what happens in the duodenum, the small intestine is absorption. All right. The only exception to that is in the duodenum where you have a lot of secretion going on. Why do you have a lot of secretion going on? Because you have this organ right here, which would be what? Pancreas. And then you have this organ up here that are secreting things into the duodenum. Okay? Because you have all of these folds, if you take out a small intestine and look at it, you'll see all of these rings that go around it. That would be these things right here. They're called valvulae convenientes or folds of Kirkring, two different names for it. On those are villi, and on those villi are microvilli. You guys ever seen that optical illusion where they have a hand and with fingers, and then hands are springing out from the fingers, and then more hands spring out from those fingers? You ever seen that before? Okay. For those of you who have seen it, actually I'll post it on Canvas if I find it. It's an interesting thing. But anyway, you have a big surface area, okay, because you have all of these folds in there. And that's important because you have a lot of stuff going through it that needs to be absorbed. So you need this big surface area for that absorption to happen. Just for reference, if we compare that to a tennis court or the surface area of the lung, it's 2 to 4 times larger surface area than those two things. So lots of absorbing power. Think of it as the difference between spilling water on your hardwood floor versus spilling water on this giant-ass shag carpet. Which one's going to absorb more? The shag carpet. Okay? All right. Each one of these villi contain blood vessels. All right, and we saw that before. We talked about the shunt, the oxygen loss that occurs in there. They also have centrally this thing called a lacteal. Now that is a lymphatic vessel, and its sole purpose in the villi is for absorbing fats. All right, so lacteal is important for absorbing fats, puts it back in the lymphatic system, and then that gets dumped into the circulatory system. All right. When we talk about absorption, we already said that the small intestine is the primary place where absorption occurs. Absorption can occur in the stomach, but usually that's alcohol, NSAIDs, and aspirin, some water too. Okay? This is why you get drunk really fast on an empty stomach. And why your friends say, I gotta eat so I last longer. All right, duodenum and jejunum. That's where most of your absorption of everything occurs. All your macronutrients, water, vitamins, things like that. The ileum, all right, we know it absorbs vitamin B12. That's really important for life. Also absorbs bile salts. All right, so all the bile salts that go to emulsify fats and end up in the small intestine go right back into the bloodstream to be recycled, and that happens in the ileum. Colon, water and electrolytes. This is why if you are in a jungle and you're dehydrated and you don't have a fresh water source, you can fill your rectum up with dirty water and not worry too much about infection by swallowing it. You ever heard of that before? All right. Now don't go home and sit upside down underneath your water faucet and try this. There are medical products they sell at stores for that. I'm joking, really. All right, rectum. Rectum can absorb drugs such as steroids and salicylates, suppositories. That's why we put them up the rectum. I remember the first time I was told as a nursing student I had to give somebody a suppository. Scared the living shit out of me. Then I didn't have to do it, and I was like, oh, God. All right, let's take a break. All right. Some things I'm going to abbreviate. This is going to be one of the slides I really abbreviate. When you absorb things, no matter where you are in the digestive system, but particularly in the small intestine, you have to go through, scientific term, shit ton of layers to get to the blood. All right. Now, what I want to mention on this is that for your carbohydrates and amino acids that are going through, you're going to need transporters to get them through, or channels. All right, that makes sense. What do we call that? What kind of diffusion do we call that? No. Facilitated. All right, some of it's active transport too, but facilitated diffusion. Fats go right through. Why? Lipid bilayer. They're soluble in a lipid bilayer. What kind of diffusion do we call that? Simple diffusion. All right, so amino acids and carbohydrates are going to go through active transport and facilitated diffusion, but fats going right through simple diffusion. All right, so fats are easier to get into your bloodstream than the other things are. All right. Small intestine, we're not going to go into this because this is more of an anatomy thing. Look at that, skipping slides like crazy. Pancreas is going to be really important. All right, so we have our tube, we have our alimentary canal, then we have accessory organs that assist the tube. All right, the pancreas is one of those accessory organs. All right, what we know is that when we go from the stomach into the small intestine, we need to neutralize acid, right? We already talked about that. The pH works around, the pH of the enzymes in the small intestine work at around 7. And we need to break down our macromolecules into the simplest possible form. All right, so we take proteins and turn them into amino acids. We take starches and, oh my God. No. Glycogen. Glycogen and turn them into monosaccharides. Then we take fats, triglycerides, and turn them into fatty acids and glycerol. All right? So they have to be broken down. Pancreas allows us to do all this. All right? So all of the chemical digestion comes from the pancreas, mostly. All right, we know where it's located. Here's that C-shaped loop of the duodenum, and then here's the head, body, and tail of the pancreas. And then we have the pancreatic acinar cells, which are going to have the exocrine function, which is what we're talking about right now. But remember, we also have endocrine cells in the pancreas. And what's the primary hormone that we get from endocrine pancreas? Insulin. That's the one we all know about. All right. So these exocrine cells will secrete juices into the tubes, and they will get squirted into the duodenum. Those words, everybody. Is anybody in here the kind of person that freaks out when somebody says moist? Squirt? Juice? Sphincter? I always found it funny that they called it pancreatic juice and stomach juice. Like, I think of orange juice, and I don't want to think anyway. So we have these openings here where these digestive enzymes go into, and notice that the gallbladder and the liver has a direct entry into the duodenum as well, where bile salts will go. All right, so that's the setup. Those are the tubes. That's how they're getting into the duodenum. And we already talked about that. All right, so what are the enzymes that get secreted? All right, first we want to talk about proteases. All right, the proteases or proteolytic enzymes, there are 3 of them: trypsin, chymotrypsin, carboxypeptidase. These are secreted in inactive form. Trypsin is secreted as trypsin-O-gen. Chymotrypsin is secreted as chymotrypsin-O-gen. And carboxypeptidase is secreted as pro-carboxypeptidase. getting ready to fuck it all up. So they're going to break down proteins into polypeptides and then polypeptides into amino acids. All right. Notice that all enzymes are secreted in excess, so we have way more than what we need, and that's to make sure that we get everything digested down to the least possible denominator, the amino acids. All right. We have so much excess that we have to get rid of 90% of those enzymes to see any problem in digestion. That's pretty amazing, I think. Or wasteful. But what happens to those proteins that are aid in digestion ultimately? They get digested, reabsorbed, and recycled as proteins, so it's not waste. Our body is very friendly to itself. All right, carbohydrate digestion. Amylase. All right, amylase. Where did we see this before? In the mouth, right? This is the same kind of amylase that's going to break down starches and glycogen into disaccharides. Right? That's the only thing breaking down carbohydrates, as opposed to the 3 proteases. All right, for fat digestion, we have pancreatic lipase. All right, naming of enzymes, right? Lipase breaks down lipids, right? Okay. Phospholipase breaks down phospholipids. Cholesterol esterase doesn't break down cholesterol. It converts it into a different form so it can be reabsorbed. Okay, so that's what they do. So 3 different fat digestive enzymes. If you have pancreatic problems, or if you're an alcoholic and you destroy your liver and pancreas, you will take all of these enzymes by pill form. I have to give these almost every time I work at my job because we have alcoholics that have just destroyed their bodies. And then when they say they don't want them, I'm like, sure you want to go down and eat then? All right. Why doesn't the pancreas digest itself? Again, we have all of these. What are our cells made up of? Lipids, carbohydrates, and proteins. So why don't these digestive enzymes digest the pancreas? Well, we gave you one clue. Every one of them is secreted in inactive form. All right. So if they're not active, they're not going to be digesting. All right. Now, trypsinogen is a little bit tricky. It can auto-activate. So we also have a trypsin inhibitor in the pancreatic cells to prevent that from happening. Like it can just activate itself. It can cleave itself. And eventually that can turn into positive feedback, but that's not what we're talking about here. All right. So here's your pancreas. When these digestive enzymes get secreted into the pancreatic duct to be secreted, they go into the duodenum and they're still in inactive form. There are cells on the wall of the duodenum, enzymes on the cells on the wall of the duodenum called enteropeptidase that will convert trypsinogen into trypsin. All right, so as soon as trypsin encounters these enteropeptidase on the wall of the duodenum, it'll convert trypsinogen into trypsin. And then trypsin can go and activate not only itself, so it can autocatalyze, but it can also activate chymotrypsinogen, procarboxypeptidase as well. All right, so enteropeptidase starts the process. That is the thing that starts the process. Then trypsin formation provides that positive feedback. So that's where that comes in. Okay, we talked about this trypsin inhibitor that prevents the autocatalysis of trypsin in the acinar cells and in the ducts. And usually, when we have acute pancreatitis, it's due to a lack of trypsin inhibitor. Could be a congenital thing or a genetic thing that you're born with, like you just aren't born with an active trypsin inhibitor. Or not enough is present. Let's say you have a mutation that lets you not produce as much of it. Or there's a blockage. Okay? Now, this same trypsin inhibitor is also found in the lungs. All right? We have where our lungs are constantly being bombarded with pollutants. Okay? So we have trypsin in there to help break apart, like even bacteria are getting down there, break apart proteins to help prevent damage to our lungs. All right? We have this antitrypsin so that it doesn't get out of control, destroy elastic tissue, leading to what disease? When you destroy elastic tissue of the lungs? Emphysema, good. And then our lungs become overly, what's the term we use for that? When they're easily inflated? Compliant, overly compliant. Good. He's like, yeah, I got this exam. All right. Really important, besides all of these proteolytic and carbohydrate digesting and fat digesting enzymes, we have bicarbonate. And the bicarbonate's primary job is to neutralize the acid that's coming from the stomach, all right? Because all of those enzymes that we just mentioned operate at a pH of around 7, all right? Around neutral pH, so we have to neutralize that. So, there's a hormone that's important for the release of bicarbonate, and that protein is called secretin. Secretin promotes the secretion of bicarbonate. Okay? That is on your list. All right. So how do we regulate these secretions from the pancreas? If we just want to secrete fluid and bicarbonate, that's dependent on the amount of acid entering the duodenum. So if we just drink some water and that gets pushed into the duodenum with acid, we're going to mostly secrete fluid and bicarbonate. Okay? The secretion of enzymes is dependent on macromolecules entering the duodenum, all right? Mostly fats, or not mostly, fats and proteins. Okay, so when the duodenum detects fats and proteins, it's going to signal the pancreas to release those enzymes. Okay? The pathways that cause enzyme release, neural, through acetylcholine, the vagal reflex. CCK, fat and protein in the small intestine, will cause the release of CCK. What kind of molecule is that? Hormone or neural? Hormone. So it goes into the blood, goes back to the pancreas, and says, hey, let's secrete some proteases and some lipases. And then the secretin is stimulated by fluid and bicarbonate, or stimulates fluid and bicarbonate secretion. Okay? So a neural mechanism and a hormone mechanism for enzymes, a hormone mechanism for fluid and bicarbonate. Secretin. All right, this is just a picture showing the same thing. I'm going to skip it. All right. We also have phases of pancreatic secretion, like we have phases of gastric secretion. Cephalic, gastric, intestinal. All right. Cephalic and gastric, both mediated by the vagus nerve in response to volume and enzyme secretion. Oh, no. By vagus, they have low volume of secretions, but a lot of enzymes. Okay, so cephalic and gastric phase, not much is going into the duodenum, but they're very highly concentrated in enzymes. The intestinal phase, once acid goes into the duodenum, secretin is released into the blood, goes to the pancreas and causes bicarbonate release. Fat and protein entering the duodenum causes CCK release to go into the blood and will cause the pancreas to release enzymes. Acid, fat, and protein entering into the duodenum will stimulate the vagal reflex and tell the pancreas to put more enzymes into the pancreas. All right? So this is just a repeat, kind of what we saw in the previous slide. They're just backwards. All right, what else did we say CCK did? So now we have two functions for CCK. What's the other function? Right. What about gastric emptying? Inhibits. All right. Why does it inhibit gastric emptying? So that these enzymes can work on the contents from the stomach. All right. So it makes sense. Like the CCK is like, hey, I got to secrete enzymes to work on these macromolecules. Stop giving me food to work on. Bitch. All right, liver. The liver. We're not going to go into the anatomy of it, but you know where it's located in the left upper quadrant. Right, Emily? Right upper quadrant. All right, 4 lobes. Again, anatomy stuff, so I'm not going to test you this on physiology. Major functions, there are a lot more than this, but it filters blood. Because remember, where does all blood that gets absorbed from the digestive tract go to first? Liver through what? Right, hepatic portal vein. Also metabolizes fats, carbohydrates, and proteins. Also forms bile, and this is going to be a really important one. Storage of vitamins and iron. And the formation of coagulation factors. In fact, most of your coagulation factors, and this is going to be important for our lecture next Friday, are made in the liver. So if you're an alcoholic and you destroy your liver with cirrhosis, you're going to have bleeding problems because you can't produce coagulation factors anymore. All right, liver circulation, about 25 to 30% of cardiac output flows through the liver. Here's what I want to say about this. Most of the blood entering the liver, 75% of it, comes from the portal system. You have 25% coming from the hepatic artery. All right, so this is bringing in nutrients. This is bringing in O2. All right, and they're both exiting through the hepatic veins to then get circulated to the body and pick up more oxygen, get rid of carbon dioxide. All right, when the blood goes into the liver through this portal vein, it's going to enter into an open system of hepatic sinusoids. Okay, and so it's almost like a lake of blood in these little hepatic lobules that are lined by these endothelial cells that will then exchange molecules with these liver plate cells or hepatocytes. Okay, so these hepatocytes are where most of the job of the liver is going to be performed. And you have this heavy blood flow that's completely surrounding all these hepatocytes so that there's a high surface area for these hepatocytes to work on. Does that make sense? Oh, and then it collects in the central vein and then collects into the hepatic, the hepatic veins, and then goes back into the inferior vena cava. Okay, in your liver at any given time, right now, let's say, for example, you have about 450 milliliters of blood. Okay, that's normal. In heart failure, where our heart can't pump as much, the liver can serve as a reservoir of blood, and it can hold as much as 1450 milliliters, 1.5 liters. Okay, so it can triple its volume as a reservoir. All right, and then if you hemorrhage or you exercise, the sympathetic stimulation can decrease liver volume by 30 to 40%. So from 450 down to 300 or so. Yeah, don't even worry about that. All right, gallbladder. Here's your liver. Here are bile ducts. All right, and here is your gallbladder. And then this is the duct that goes down into the duodenum. All right. Our liver secretes a lot of bile each day. All right. Its job is fat emulsification. So it takes big globs of fat and turns them into little globs. Okay. And what we effectively do when we do that is we increase the surface area for enzymes to work on. Okay. The gallbladder will take most of this bile that the liver secretes per day, concentrate it, and store it in this little sac. Okay? So it'll concentrate it and store it in this little sac because you really only want bile secreted when you need it. It's not necessarily bad if there's a continuous flow. In fact, does anybody in here have their gallbladder removed? No? Some people do. They live perfectly normal lives. Why? Because the liver can still provide that bile. It's just a continuous flow instead of regulated flow. All right. So it stores bile. I'm not going to go into the details of that. But the secretion of bile occurs in two stages. First, we have the hepatocytes. Remember these hepatocytes, and here's our cell that's in contact with the blood here. Hepatocytes, they will secrete contents of bile into the bile duct. They'll go here to the bile duct, and then the bile duct will then secrete water, sodium, and other things that the bile needs to be able to function properly. All right. Mainly, the water that it's secreting into the bile duct will allow the stuff to flow, right? If you just you guys will get this next week when we talk reproduction. I always give this example of reproduction. Everybody, people who are not educated in sexual reproduction say that you ejaculate sperm. So no, you ejaculate semen. If you just ejaculated sperm, there wouldn't be any volume to it. It might be just a poof. So, same concept here. You don't want to poof into the bile duct. You want good flow, so you add water. All right, I am not going to test you over all the constituents of bile. You're welcome. But this gives you an idea for what's in there. The most abundant solute in there are the bile salts, and that's what's going to have the digestive function. The other thing that you'll have in here, though, is this bilirubin. Bilirubin, as I was talking to some guys that group there the other day, comes from breakdown products of red blood cells. Okay, that bilirubin is what gives your poop its color. All right, so the bilirubin is what gives your poop its color. So that comes from the gallbladder, goes in and gives your poop its color, and that's how we get rid of red blood cell byproducts. It's also why your urine's yellow. Anybody know what disorder we have when we have too much bilirubin? Pardon? Jaundice. All right. Bile salts are amphipathic, which means they can interact with both lipid-soluble substances and fat-soluble substances, and that's why they're good at emulsifying lipids. So they'll take these, again, big globs and turn them into smaller globs that the digestive enzymes can work on. Okay? I know I'm not going into a lot of detail about this. That's not really that important. But we're trying to build a case for why these work as bile salts. They provide this function. Okay? All right. The gallbladder empties, right? Remember, it's the liver secretes, gallbladder concentrates and stores until the body needs bile. Since bile is involved in the digestion of fats, then fat entering the duodenum will be a signal for gallbladder contraction. And what do we know is released when we have proteins and fats entering the duodenum? CCK. So a third function for CCK. Decreases gastric emptying, causes the pancreas to secrete digestive enzymes, and causes the gallbladder to contract, squirting bile into the duodenum. Okay, so all of those are then going to get mixed together and digest your food. All right, excuse me. Liver processes cholesterol. It uses cholesterol to synthesize bile acids, so that's one way we get rid of them from the body. Okay. Cholesterol secreted into bile, so we're actively getting rid of this at 1 to 2 grams a day, which is a lot if you think about it. Although it goes to make bile salts, the actual cholesterol in the bile has no known function. That doesn't mean it doesn't have a function. We just don't know what it is. But if we have too much cholesterol in our bile, what can that lead to? Do you guys know? No. It's in the bile, so it's not anywhere in the body. It's being squirted out of gallstones. So too much cholesterol can lead to gallstones. One of the mechanisms by which it can lead to gallstones. Okay. Excuse me. All right. Some secretions by the small intestine. So all we've talking about right now is breakdown of stuff. What about some secretions? They secrete an alkaline mucus. Okay, so it's mucus with bicarbonate in it. That's stimulated by the vagus nerve and inhibited by the sympathetic nerve. That makes sense. So the more vagus impulse we have, the slipperier our intestines get. Okay, and then we have these crypts, libercune, that's how that's pronounced, libercune, that secrete watery fluid at 1,800 milliliters per day, again, to increase volume and help things move along. All right, absorption. We're in the home stretch now. Absorption. We've already started talking about this. Okay? Small intestine, the most important absorption, it absorbs everything. Okay, small intestine absorbs everything. We've already talked about that. Carbohydrate digestion. Where does it begin? Mouth with amylase. Where does it carbohydrate digestion end? Small intestine with absorption. Okay? Due to the functions of amylase. And I will also tell you, remember amylase breaks down glycogen and starch? Pretend that's a sugar. Into disaccharides. But the wall of the small intestine has enzymes that will break these into monosaccharides. And monosaccharides are absorbed. All right? That's where we come into the lactase, maltase. All right, protein digestion. Where does that begin? With pepsin and acid, right? You need that acid to activate the pepsinogen to pepsin. Where does it end? Small intestine. Through the actions of Which enzymes? Begins with T, rhymes with ripsin. Trypsin. Chymotrypsin and carboxypeptidase. Right. And then we absorb them and they're carried away into the blood. Absorbed in the villi. All of this stuff's absorbed in the villi. Fat absorption. Fat digestion begins where? Small intestine. All right, we haven't talked about it anywhere except in the small intestine. So we have what's emulsifying it? Bile salts. What's breaking it down? Lipase, phospholipase, and that other one that works on cholesterol. Right? Remember? It's cholesterol esterase. All right? Are they absorbed into the blood? Fats. Lymphatics. Those lacteals that are in the villi. Good. All right, and so this is kind of how that works. Fats, they go right through the cell, right? And how do they go through the cell? By the process of? Thank you. Love that. I love Daniel. I'm not making fun of him. It's just really, I love how he says that. Thank you. Okay. So notice they're absorbed as small particles. In the cell, they get packaged into these things called chylomicrons. Have you heard of VLDL, LDL, and HDL? Chylomicrons are a step above VLDL. So very low density lipoproteins, Chylomicrons would be super mega ultra high density lipoproteins. Low density lipoproteins. Okay? So they have a lot of fats in them. They get put into the lymph and then transported into circulation. Okay? Where they would get dropped off at certain cells, get processed by those cells, by the enzymes there. Then they get turned into VLDL and LDL, not HDL though. I don't care about the molecular mechanisms of that. Just want to let you guys know what happens. All right, absorption. So when they enter the lymphatics, they empty into that cisterna chyli, which is right between the cruri of the diaphragm, right behind the aorta. Okay, that's where the cisterna chyli is located. And then makes it up into the thoracic duct, and then subclavian vein into circulation. Okay. All right, now into the large intestine. All right, here's our ileum going in. We have a valve right here, and then we have the cecum, and then the ascending colon. All right, what's this valve called? Ileocecal valve, or ileocecal sphincter. I'm going to keep that in mind that you don't like sphincter. All right, the major job of this large intestine to absorb water and electrolytes, we talked about that already, but it also forms and stores feces. So it's our recycling center, or no, it would be our Republic Services. Never have your garbage picked up by them, by the way. They are, my bill went from like 150 to 350 in one cycle. We called them and were like, no. I didn't. My wife had to do it because it's her house. All right. So here's the cecum right here. Here's the ileum, ileocecal valve. It's a pouch that kind of like if this is ground zero, 1, 2, 3, 4, 5, 6, this would be like -1 as far as anatomical relationship goes. Attached to the cecum is the vermiform appendix, right? And there's a lot of argument over what the actual function of that is, but one of the things we think it does is it harbors bacteria And when you have like something that eliminates all the bacteria from your gut, it will replenish it. That's one of the supposed functions of it. Okay? Ascending, transverse, descending, S-shaped, which we call sigmoid, rectum, and anus. Okay? So that's the whole path going through them. And of course, in the rectum and anus, we have sphincters here as well that regulate the passage of fecal matter. What are these things called? Pardon?
Are they hostra?
No.
The actual
No, these specific humps right here and here. All right, which one is this? Hepatic spine. Good. You guys are awesome. You're going to kill that midterm. All right. So we know that the large intestine absorbs water, right? As it absorbs water, what happens to the contents inside of the colon? They get drier. All right. So as chyme is entering the large intestine here, you absorb water, you absorb water, you absorb water. The contents here should be fluid in nature. So if you put a stoma in here, what's going to come out is fluid. Right side. Goes through here, you lose more water. Contents in here are mushy. Which, if you're talking about this, it sounds really gross. All right, so the contents of here will be mushy. Put a stoma here, you're going to get mushy stools, like applesauce or yogurt. Maybe a little bit thicker than that. Splenic flexure, semi-mush. So they're starting to form now. Get rid of more water and more water. In the descending colon, semi-solid. Going towards the sigmoid colon and rectum, solid. All right, so you're losing fluid along the way to reabsorb it back into your body. Okay? Does that make sense? This might come up. I don't know if they talk about this because I don't know fully what they talk about in all the Clin Med, but they may talk about different digestive disorders and stomas and how you might, what you might expect from them. So this is an introduction to that. Okay, important note here. If we have things rushing through the colon too quickly, then we don't absorb as much water and we have diarrhea or loose stools. If you've ever said, all right, I need to drink a lot of water, but I need to drink a lot of water, but I'm going to drink it all right now. Later, not too far later in the day, you're going to have loose stools because you just drank too much and you just flushed your system out. Which can be good unless you, let's say you have a date that night. That'd be bad. All right. Ileocecal sphincter. Really important. That's right here. You guys told me that. That is a one-way valve. Okay, it's a one-way valve. And which way does it go? Left or right? It's a one-way valve to the left. So when I put things in here and then I start contracting, I'm distending, right? I'm increasing pressure. Are things going to flow backwards? No. Why? Because you're contracting this ileocecal sphincter. One-way valve. You put things in here, but then you increase the pressure here. They don't flow backwards because this closes. Okay? There is longitudinal smooth muscle, the tenea coli, rather. Ascending, transverse, descending. Those are smooth muscles that the circular muscles around here latch onto because the large intestine isn't really attached to anything. There's nothing for it to, in order to move things, you got to pull, right? These act as a mechanism for the circular muscles to pull against. And that's why you get these little pouches called haustra. Okay? Once you go all the way through, you get to the rectum and the anal canal. And within there, you have the internal anal sphincter and external anal sphincter. All right, you have two sphincters. One is under involuntary control. One is under voluntary control. And it's the training of the one under voluntary control, the external anal sphincter, is what occurs during potty training with kids. Okay, we're training this external anal sphincter. We're training them to recognize and then whoop, clamp. All right, we talked about this. The only thing I want to say here, the large intestine actually doesn't absorb water. It absorbs electrolytes. And anytime you're absorbing a large amount of electrolytes, what follows? Water. All right, so it's a passive process. All right, and of course we know we have a lot of intestinal flora. We talked about that. It can be replenished by the appendix, but we also need these bacteria because they break down products that our body can't, such as cellulose. All right, they're not efficient enough to digest that whole kernel of corn that appears in the toilet the next day, but they can digest cellulose. They also produce vitamins that we need, and they are responsible for that gas smell when we flatulate. All right, of course, you have mixing movements that break the fecal matter into segments. You could argue that it doesn't do that very efficiently because sometimes you get this like snake in the toilet. Don't say you've never seen one. When you have to go to the bathroom, you have peristaltic waves that occur in 2 or 3 mass movements. So when you're sitting there and all of a sudden you feel like, ooh, gotta go, that's a peristaltic movement. That's moved things to the internal anal sphincter, stretched it, and made you go, ooh. So sensory input to the brain, making you say, all right, clamp down on that external anal sphincter. You guys don't think about this. All right, when that happens, you have the defecation reflex, and we'll talk about that on the next slide. We are almost done. When we void or poop, what ends up in the toilet? Or anybody here a runner? And when you're in a long run and you have to go in the woods, you have to go in the woods. That's how I got poison ivy one time, all in here. And I ran a marathon like 3 weeks later. Anyway, I'll tell you about the trauma of that later on. All right, so what's in fecal matter? Water. There's still some water there left because it is mushy. Electrolytes. You didn't get rid of all of them. Mucus, because you have to slide it through. Bacteria, because they're not all, some of them may be hanging on for dear life, but they're going to go on with the fecal matter. And bile pigments. Right? Remember I said the bile pigments from red cell, blood cell breakdown is what gives it its color, so those are going to be in there as well. In fact, I proved this to my kids once. This is kind of bad I am. I said, your poop is not actually brown. I said, next time you poop, wipe and tell me what color the toilet paper is if there's not like a smear on there. It's yellow. It's yellow. All right. So the intrinsic defecation reflex. All right. What does this mean? Does it need nervous system input? No. All right. It's all within the in the bowels itself. All right. It's initiated when feces enter the rectum. What happens during that time? We have distension. Right? That's the signal. Okay? Rectal distension initiates different signals that spread through the myenteric plexus in that area, and it causes contractions that force fecal matter downwards. So you're going to contract here and force fecal matter downwards. All right, towards the anus. All right, the internal anal sphincter will relax. Okay, so this internal sphincter right here will relax and allow fecal matter to exit. Right? The external is voluntary, so you control when that relaxes. This is why the internal is like, hey, we got to go. External saying, not yet. I got to get somewhere. Okay, so that's voluntary control. You also have a spinal cord defecation reflex. So this has to go through the spinal cord to happen. What this does is it does the same thing, really, but it just intensifies the reflex. Okay, so let's say you're on a walk, and then it hits, and you're like, ooh, okay, got to go. And then you're almost home, and it's like starting again. You're like, oh! That would be the secondary reflex. They say as you get older, you start talking about your bowel movements more. I turn 47 next Friday. I guess it's true. So anyway, this happens through sacral nerves, through the spinal cord. Okay? Again, parasympathetic fibers are going to modulate this. All right. Higher brain centers, we can modulate our defecation reflex. When we want to push harder, we can take deep breath, Valsalva maneuver, closure of our glottis, increase abdominal pressure, like when you're on the toilet and you push, that's what's happening. That moves fecal contents downwards. That's like a pressure. You're pressing on all of the intestines and pushing things downwards. If we have an injury to our spinal cord, that can make defecation difficult. It can still happen, but it doesn't occur as easy. So you need that spinal cord reflex to make things go out. Okay? The cord defecation reflex can be excited either digitally. So when your child is not pooping, you can stick a finger up there and wiggle around a little bit, and sometimes they will go. Or with an enema. So you squirt things up there, they expand the rectum, and then you can go. Okay. Two assholes standing next to each other. Sorry, I had to. All right. So there's your rectum. Here's your anal canal. This is your internal anal sphincter right here. This is the external anal sphincter, just to kind of give you an idea for where they are in relation to it. They call this internal because it's internal, like it's just on the outside of the mucosal lining, whereas the external is a little farther out. And notice it's a much bigger muscle group because one, it's skeletal, and two, you need a bigger muscle group to control that. All right? Your body wants to get rid of something. It takes a much bigger force to control you not getting rid of that something. All right? So, I believe that ends everything. That was awkward. And I wear this shirt specifically for today. That's related to this lecture. So, you guys have a great weekend. If you have any questions about this material, feel free to email me. I think I have to work nursing Sunday, so I won't be able to answer, but I'll get back to you as soon as I can.